Good evening, students. The other day we discussed the general characters of bryophytes, and they are the first embryophytes because after gametic fusion they produce embryo, and also they are the amphibians of the plant kingdom, and we discussed those characters in that. Now let us see the occurrence. Today we see the occurrence of these bryophytes, where you can get the bryophytes. So now regarding the occurrence, generally all bryophytes found in moist, damp and humid conditions, and very few are water plants. Those are called generally hydrophytes. The plants which grow or which float in water are called the hydrophytes, for which the examples of Rixia flutens, Rixiocorpus natans, Riella, etc. So they are floating in the water. So here you can see the Riella is floating in the water, and another, the Rixiocorpus, this is also floating in the water. So these are hydrophytes. Next, some plants or grow on moist rocks, shaded grounds, tree trunks and other similar moist places. Few species grow as bogs, that is, they grow in soft, wet, muddy areas. So generally the plants which grow in soft, wet and muddy areas are called bogs. For example, sphagnum. You can see this plant, it grows on muddy areas where you can find in a black color, there you can find the place where it is living, that is a muddy place. So this is a bog moss. That's why the sphagnum is also called bog moss. Next, some species, sometimes what happens, many species of these bryophytes are epiphytes. The plants grow on other plants. Plants grow on other plants. Those are called the example as a epiphyte example, Dendroceros. So there is a branch of some plant. On that plant, these plants are there. These plants are called. This is a Dendroceros plant which grow on branch of another plant. That is so these are called epiphytes. Very few or very rarely some species are A.V.V.S. That is, the plant which grow on the leaves of other plants is known as A.V.V.S. plant. So growing on the surface of leaves of other plants. Example, Radula protensa. These plants grow on the leaves of other plants. So the plant which grow on other plant is called epiphyte, or the plant which grow on the leaves of other plants is known as A.V.V.S. plant. Phylla means leaves. So here you can see that is given a rounded red rounded circle is given to you for to see the plants are growing on the leaves of other plant. Leaves of other plants, plant is growing on the leaves of other plant. So this is epiphyllous plant. Example, Radula protensa. Next you see the classification of bryophytes. So as with other classifications, Bryophyta is also classified by many bryologists. As I told you already, the science of study the bryophyte is called bryology, and the scientists who study are bryologists. So here the Smith, Rothmeiler, Reimer are classified the bryophytes into three classes. Those are Hepaticopsida, another is Anthoceratopsida, third one Bryopsida. The Hepaticopsida plants looking like liver, those are called liverworts. They are looking like liver. The leaf, the thallus is just looking like a liver, so these are called the liverworts. And the next one, Anthoceratopsida, these thallus, the sporangium is a horn-like structure, so that these are called hornworts. The third one is Bryopsida. These are developed bryophytes, so these are called the mosses. In general, bryophytes are called mosses, but because of this one, so the advanced bryophyte is moss. It is under Bryophyta class. Now let us see the characteristics of each class. The first one, Hepaticopsida, the liverworts. The gametophyte is dorsi-ventrally flattened. Just I told you on the liver, this is dorsi-ventrally flattened structure and commonly known as liverworts. Thallus is attached to the substratum by unicellular rhizoids, which may be smooth or pegged rhizoids. They may be smoothed or pegged rhizoids. If a rhizoid is like this, that is a smoothed rhizoid. If the rhizoid having peg-like structure inside the root hair, so these are called the pegged rhizoids. Pegged rhizoids both you can find in some species. Next, the gametophyte is internally simple, made up of many tissues, whereas here in this Hepaticopsida, the internal tissue of the thallus is differentiated. One is called the photosynthetic region, another is the storage region, like that. And photosynthetic cells contain chloroplasts without pyrenoid, and air spaces are present for gaseous exchange. The photograph you will see later. This is the photograph you can have. This is the transverse section of the thallus of Rixia. In this you can find these are some are smooth rhizoids, some are pegged rhizoids we have, and it is differentiated into internal structure, differentiated into two regions, and this is called the element. This is assimilatory or photosynthetic region, and the lower region, this is called the storage region. And these are here photosynthetic filament like structures are formed, and the end of the cell is not having chloroplast, and they form as a upper epidermis. In this, in between, you have air pores are there. These air pores help in the exchange of gases. Air pores in the epidermis like structure, exchange of gases. Next, regarding the characters, the sex organs develop on the dorsal side of the dorsal side. Dorsal side, if you happen to see the internal structure here you can have, this is the dorsal side, this is the ventral side. The ventral side having these rhizoids, the dorsal side you have these assimilatory region or photosynthetic filaments. So it develops, the sex organs develop in the dorsal side of the thallus. And the development is superficial, not from inside, superficial cells, that is called exogenous development. And the antheridia are generally the male sex organ, and the archegonia is generally called the female sex organ. They develop from single initials, single cells from one cell. Next, here you have given the antheridia and archegonia, where you can find archegonia and antheridia. Here antheridia is developed in the here, and it is the archegonium from the superficial cells of the dorsal side. And in the antheridium you have a sterile jacket layer around this, and inside you can find androgonial cells, and they divide and give male gametes. Here the archegonium, it is a flask shaped structure, and it is having a broad venter, and a neck is there. At the tip of the neck there is a neck, these are the cover cells, and the neck is having neck cells. In between the neck you have neck canal cells, and in the venter, in the broad portion, you have a venter canal cell and a big egg cell. And at the time of reproduction, the male gametes, a number of male gametes show this are called chemotactic movement, that is moving towards a chemical substance and reach the archegonium. A number of male gametes enter into the archegonium. Finally, one male gamete fuses with the egg and form a diploid zygote. A diploid zygote, that is inside the thallus only, inside the archegonium, the thallus is developed. The sporangium is developed inside the female thallus or inside the archegonium. So the sporangium is simple and differentiated into foot, capsule, etc. Look here. Here, after the fusion, a diploid zygote is formed. That diploid zygote is developed into sporogonium, and the sporogonium is differentiated into foot and capsule or foot seta and capsule. Here you can have this sporogonium having a foot from which it absorbs food material from the gametophyte, and a stalk like structure This is called seta. At the end of the seta, you can find a capsule, that is the sporogonium. So that it has sometimes only two parts, foot and capsule only, the seta may be reduced, or sometimes you have both three parts you can find. So now we can have difference here, whereas here you can see one foot and capsule, only two parts are there here, whereas here you can find the foot, seta and capsule, both are there, and three structures you can find here. So here inside the sporogenous tissue is differentiated to spore mother cells. They are diploid, they undergo reduction division and form haploid spores. So each spore undergoes a reduction division and gives 4 cells. These 4 cells are arranged in a tetrahedral manner. In the tetrahedral means in one side you will get only 3 spores and the remaining spore you can find in the opposite side. So these are called the tetrads. After completion of this tetrad and the spores are liberated, after finding a suitable place, they develop into gametophyte. So these spores lack green cells and stomata. The sporogenous tissue gives rise to spore mother cells in Rixia, are spore mother cells and elaters. So elaters are sterile, hygroscopic with spirally thickened walls, help in the spore dispersal. Spore dispersal. So here you can find spores only, the spore mother cells and spores only, spore mother cells. Here you can find the sporogenous tissue and elaters. These are the elaters, spirally thickened elaters. They help, they are hygroscopic, absorb water and liberate the spores. These are called elaters. So this texture you can find in Markantia, whereas normal spores here mother cells in Rixia and elaters develop in Markantia. The Rixia is homotholic, that is both Archegonia and Antheridia develop in the same thallus. Example Rixia robusta. Are heterothalic, they may develop on different thalli, that is very rare. Example Rixia discolor. Another example in this hepatic opsida is Markantia. It is always dioecious, that is male and female thalli are separate. This is, you can find, this is the male thalli. There you can find sporangium, it's just like a star-like structures having a stalk, and here you can find this is a female thallus. Okay. So next you can go to the next class, that is Anthoceratopsida. In the Anthoceratopsida, the members are generally called hornworts. As I told already, the sporogonium is a horn-like structure on the thallus, so these are called hornworts. The gametophyte is dorsiventrally flattened thallus without internal differentiation of tissues. In the previous, you can have hepatic opsida, the thallus is differentiated into the dorsal storage region, here you can find the ventral storage region, and there another, that is upper side, that is the dorsal side, this the photosynthetic region. But whereas here no differentiation, similar cells you can find. And each chloroplast contains one compound pyrenoid, whereas in hepatic opsida no pyrenoids. Here you can find pyrenoid. Let us see the diagram. So here this is Anthoceros, this is the thallus, and the sporophytes are just like horn-like structures, so this is called a hornwort. This is the gametophytic tissue. Next, this is the internal structure. I told you already, the internal structure, there is no differentiation of tissues, but here you can find some unique characters. One, that is the chloroplast having pyrenoid. Another, here you can find the the blue-green algae are also found in the thallus and give this nitrogen material. And these are the starch grains are found, and these are the unbranched unicellular rhizoids. Next, the sex organs are embedded in the thallus. They develop from the dorsal hypodermal cells. Origin is endogenous. So in Dixia, that is exogenous, and whereas here from the inner cells, so it is endogenous origin. And the sporogonium is slender, elongated, and consists of a foot, a meristematic region, and a long cylindrical capsule. This is very interesting, and so far the word meristem is not used, but here in this sporangium of or sporophyte of Anthoceratopsida, the meristematic tissue is there. It gives continuous growth, whereas in the previous one the growth is limited, whereas here it has a continuous growth because of the meristematic cells, meristematic region. So the capsule grows by means of intercalary meristem. Since the meristem is found in between the cells, it is called intercalary meristem. The growth is indeterminate, indefinite. The capsule wall is many-celled, rich in chloroplast and stomata. So hence they are autotrophic. So they can prepare their own food material because stomata is there and the chloroplasts are there. So it may be the independent. Sporogonium is two-layered, outer amphithecium and inner endothecium. Generally it is common for all sporogonium. The outer wall is called amphithecium and inner endothecium. The archisporium is derived from the amphithecium inside, whereas the sterile columella from the outer endothecium. The archisporium gives rise to spore mother cells and pseudo-elaters. No bands on the elaters, elater-like structures, no bands. So now you see the sporangium, the sporophyte is very interesting. There you can find the sporophyte having a foot-like structure, and you can have a just like what's called a horn-like structure, the full sporangium. So where you can find, because of the sporophyte, this is a what's called you can have this meristematic tissue. Meristematic tissue, it grows continuously. It gives sporogenous cells, and sporogenous cells undergoes spore mother cells. Spore mothers undergo meiosis and form haploid spores. So every stage you can find, and then it will be break into two halves and release the spores. So these spores you can find from the different stages. The section is given here. Sporogenous cells and spore mother cells undergo reduction division, form haploid spores. Haploid spores are released out. So in the same way, this is a speciality that you can find in Anthoceras. The third class can have this Bryophyta. This is developed bryophytes, where bryophytes are commonly called masses. These are called generally masses. The gametophyte is well differentiated into stem-like, leaf-like and root-like structures. So far in both Anthoceratopsida and Hepaticapsida, the thallus is dorsi-ventrally flattened thallus. When coming to the Bryophyta, a little advancement where there is a stem, there is a leaf, there is a root, but not true roots, so true stem. So these are called stem-like, leaf-like and root-like structures. Rhizoids are multicellular, branched with oblique septa. Leaves are green, papery with a midrib. So here you can find the diagram. There you can find the leaves are there, and the leaves having small midrib, and they are spirally arranged on the stem like structure. So regarding the characteristics, the stems show central conducting strand that is made up of parenchyma only. The remaining normal cells, also some group of cells in the middle, they help in the conduction, but they are only the parenchyma cells. In the leaf also, at the midrib region, you can find a group of parenchyma cells attached together. The sex organs develop from the apex of the stem or lateral branches. The lateral branches, there is separate antheridial branch and separate archegonial branch. So here you can find the antheridial branch, this archegonial branch. In between you can find short absorbs like the paraphysis are there in between this antheridia. They give support or use as called stabilizing substance or absorbers or shock absorbers. The sporophyte shows differentiation of foot, seta and capsule. The sporogenous tissue develop from the outer layers of the endothecium along with the central columella part is there that you can see later. The wall of the capsule consists of several layers of chlorophyllous cells and stomata. So, these are also autotrophic. They can prepare food material by own, not depend upon the gametophyte. The capsule is a complex structure and differentiated into the following three regions. So, a sterile basal region, generally if you happen to see the capsule, I will show later, this is the capsule. And in this capsule, the lower portion is called apophysis and the middle portion is called theca, and here you can find that there you can find this peristome teeth and also operculum. Operculum is nothing but a lid. Lid is called the operculum. Below the lid you have some teeth-like structures, those are called peristome. So, total three structures you can find. The sterile basal region, because sterile means there is no development of spores, and the fertile region in the center only you can find the development of sporangia. So, fertile region is called the theca, and the upper region consists of the operculum and peristome. These are the teeth like hygroscopic help in the dispersal of the spores, because when the sporangium matures the lid may be removed and the peristome teeth comes up and help the tree to disperse the spores in a what's called installmental way, not at the same time, at different places it will release the spores. So, this is the structure I told you. This is the capsule and it is having a seta, capsule seta is there, and this is the total capsule like structure. The lower portion, this is called apophysis. So, there you can find in the apophysis the sterile region, and then this is the theca proper, where you can find two, here you can find two groups, here one group, in another group, these are having the sporangia. This sporangia having the gaps, air spaces are trabecular there for supporting purpose, just like absorbing or what is called stabilizing substance, like that. And inside you can find the spore mother cells. They undergo reduction division and form haploid spores. Here you can find the rim and operculum, that is the lid. Inside the lid you have peristome teeth. These are all peristome teeth. And the central sterile region, that is called columella, it also helps in the liberation, and also the peristome teeth also help in the liberation of the spores. So, these spores, after finding a suitable place, they develop into the funaria or moss gametophyte. So, these spores germinate to give a protonema like structure. Protonema is nothing but a filament like structure, and this is having sometimes some of the buds are present here, and each bud is developed into a gametophyte. Each bud is developed into the first stage is protonema stage, a branched filamentous form, and then it is having buds are there. Each bud develop into that particular bryophyte. So, example funaria and polytrichum plants. Example funaria and polytrichum. So, here you have, this is the funaria and this is the sporophyte. The upper one is the sporophyte and lower one is the gametophyte. Sporophyte and gametophyte. This structure you have this polytrichum plant. Polytrichum plant. Right. Next, we will see the spores germinate to give a protonema stage from which several gametophytes develop. Next, let us see, just like algae and fungi, let us see the economic importance of bryophytes. Regarding the economic importance of bryophytes, bryophytes form dense mat over the soil and prevent soil erosion. So, there are very small plants and having a bunch of rhizoids are there, they bind the soil particles, and some way it help the prevention of soil erosion. The second one is the property of conserving moisture, because the plants absorb moisture for a long time. These plants are used as packing material for cuttings and also seedlings. For small plants or plant cuttings, they used to use this bryophyte members because they absorb moisture and they retain moisture for a long time, so that they are used. Some bryophytes, for example sphagnum and other mosses, are used as packing material for fragile goods, glassware and standard quality of crockery, because after drying what happens, they are like soft what's called as soft leaves, so they are used to pack the glosses and also fragile very essential materials and glassware and these cups and etc., finger cups etc., they are stored or also packed with the help of these sphagnum like bryophytes. Another important thing, bryophytes like sphagnum have an antiseptic property, so that they are also used as bandages on wounds. On wounds etc., it is used as a bandage material, the sphagnum. So according to this Bryophyta, the sphagnum having very important uses you can find. Another very important thing, the mosses like sphagnum, Hagnum, which accumulated over the years on the compressed mass, generally in a particular areas where the sphagnum is going years together, they dry and mixed with soil, and again you can have form into just a land like structure, just like a land in a particular area where it is compressed and passed, that is called the peat. So this is called the peat. So sphagnum has got many names. One is bog moss, another is called the peat moss. After drying, it is used as coal. So it is used as just like a coal, it gives energy and as a coal. And another thing, mosses are the good sources of animal food and in rocky areas. In rocky areas where some of the algae are used as food, animal feed or fodder. So regarding the peat moss, here you can find this bog moss is accumulated. Bog moss is accumulated, see, accumulated just like a form area, full area of land form as peat. So this peat on firing, it is used as coal to get energy. Okay. Thank you.